is a sample jar you didn't know existed.
That's right.
Yeah,
It's filled with
I'll
tons
just
of
keep
cool
my eye
stuff.
on this jar.
It's
And,
Mystery Jar 8.
Yeah, mystery jar.
It's like, yeah.
It's. I swear, it's the same place that the socks go.
Or maybe
Yeah.
or it's the,
All right.
it's the,
All right.
it's
Megan,
the chamber
I'm gonna
that,
give you your camera
it's
back.
the chamber for the
Cool.
samples they sent to
I want to look at
Hogwarts.
this
It's like
coral.
chamber 5B.
Sorry, video. I'm still extending, so
Oh,
that
okay.
might affect your.
Yeah, all good. Are you all done extending, right?
Yes,
Okay,
I
sweet.
am all the way out now.
I'm gonna zoom in as much as I can.
And here I can tilt pan.
Oh, perfect.
Thank you.
Nice job
Cool.
setting up, Megan.
It's a brittle
It's
star?
a sea pen
Oh, see?
with a snake star on it.
But
Snake
not an
star.
umbellula.
But it's not an
Oh,
umbellula.
oh, sorry, Gabby.
That's a nice one. All right, thank you. We can zoom out.
Okay, zoom it out.
I really thought this was going to be less down than it was, just based on the map, but
It
here
was
we
a
are.
lot of down.
It was a lot of down.
I wouldn't have anticipated that.
Yeah. Okay, I'm going to start the ship up again.
Yes, thank you.
Bridge nav.
Sorry.
Okay,
that's me.
let's go with a 30-meter move, 30, at 045.
It really
A
is
30-meter
speed.
move at a 30-meter move at 045, 0.3 knots.
Ah,
Um,
I thought you were taking us to 0.45 knots there for a moment.
really?
Ramming
I was trying to
speed.
think about too many things at once, and it all came out in a jumble.
Yeah.
That soft
hard.
sediment, it's the tempting thing. You can fly. It's the salt flats.
Yeah.
So,
Squishy.
Rye, can you write down in the red book that there might be a starfish somewhere eating everything?
As long as it's not in with the bamboo coral, it should be fine.
Okay.
Because that's what they eat.
Yeah, we have not
Oh, no.
rotated into any of the jars that have coral in them.
Okay, so we want to
Interesting.
keep, we don't want to rotate in,
Clockwise.
yeah, we don't want to rotate into any of those jars. Data, did you catch that too?
Yep, got
Okay.
that. Oh, so you're not going to rotate anything into 6 or 7?
We'll keep, we won't, we'll only rotate into 6 or 7. Those
Right,
are the
got it.
only
So
ones I think
presume?
that are safe
Okay.
for a starfish to climb into.
Yeah.
Oh, that's cool.
It's a little brittle star.
And
Very nice. This is a ophiacanthid brittle star. We can zoom out.
Okay, zoom it out.
Usually we see those like on things, but it's just hanging out all by itself. Oh, there's rocks coming up.
Yay.
Yay.
Rocks means biology.
There they are.
We've got a really big problem because there was Gabby Nav and there was Gabby, I don't know.
Pilot.
Yeah.
And now you're Gabby Nav.
Now
Gabby
we've
Nav Jr.
two Gabby Navs.
there can only be one Gabby Nav.
Yeah, it's true,
You guys
and
got
I'm
it.
not it. So here we are.
Back to the rocks.
Yeah, so it seems like we might be finished with the saddle.
Yay.
Yay.
Yeah, back to rocks, yeah. Chris is stoked.
Well, there's more stuff happening on rocks, right?
There usually is. But I've definitely been to
mean-
some sediment places where
I
it was
mean,
happening.
Once you've like Once you've looked at current direction and current speed from the size of the ripples, there's only so much you can do with looking at sediment. If you can't wiggle your toes in it. And since I still got a sleeping bag full of broccoli, don't even feel the need to do that anyway.
I feel like I'm gonna like pull a broccoli joke on another cruise and
no one's gonna get it.
Yeah.
Just only Megan is gonna laugh.
I'd laugh
If you were there.
Yeah.
if I was there.
Well, you're all
Well,
gonna
we're
be-
all
I'll
gonna
make
be
it
on
be,
Palau,
like-
so you know maybe we
You
just
only have
do
to
the
wait
same
till
watch
Palau,
again.
right?
Yeah.
Yeah.
Just see if you remember by Palau.
You're on for 2 weeks?
So many
far?
vegetables
Yeah,
under the
I'm
bridge.
on for 2 weeks of Palau.
Nice.
So what are those dates?
October 26th to November 8th, maybe?
And all three of you guys are going to be in that expedition as well?
Yep.
Yep.
Yeah, I'll be-
Yeah.
Interesting.
You got the dream
We're
team
gonna
in
get
the
a-
front
gonna have
row.
We're
The
gonna have to
dream
get,
team.
our chat on.
Yeah. Oh, yeah.
See if we can like sneak in some questions into that.
Yeah, I want to be hearing from Chris German while we're out here again.
Do you always serve on the same watch as each other, or will it be mix and match and you'll be with other pilots? And
It often gets mixed up.
Yeah.
It's really like they just try and optimize for the crew that they have out there. They try and match like navs who know the system with pilots who don't, and vice versa.
I feel like, there could be like school principal rules of like, if anyone has been listening to this one, like those three have way too much time together.
Yeah, oh,
Like,
there's-
I think they should be separated.
There's
Yeah.
certainly
Uh-huh.
that absolutely
you're creating
happens.
havoc
Yeah,
Yeah.
in the live feed.
Or
Yeah.
maybe it's just, it's on the 12 to 4.
yeah. Sometimes you like
If you're a
put in.
science lead, you don't have to know. If you live on the 8 to 12, it's
Yeah,
like,
exactly.
I'll just go to bed and like.
ROV lead always lives on the 8 to 12, but
Yeah.
like you build the watches for chemistry, for experience, for, I don't know, generally people having a good time and getting the work actually done.
Yeah.
We got some people asking about what's this broccoli thing we're talking about.
You
Yeah, if
don't
you
wanna
missed
know.
the first dive ever with Chris, then you'll have no clue what's going on.
It just had to be there.
It just started with what was your favorite thing you've eaten on the ship?
Yes,
Wasn't
yeah, that was a question.
Had to watch the pilot episode, unfortunately.
Well, once you know that part of like, I really liked the broccoli that they were serving that first week, you can make up any story you like and it won't be any more absurd than mine. So.
But we haven't had broccoli
look.
for a while now. It's very
No, one of the
No,
things
it's
that's
all
gone
gone
missing.
now.
yeah.
Look at those tire tracks.
Yeah.
That's a big earth mover came through here. Huh? What kind of size sea cucumber would that be? I haven't seen Dune 2 yet, but
I haven't either. I want to see it though.
I think this might be it. We maybe just walked onto the set.
Yeah.
Yeah, you can really see the current down in these like deeper regions.
Mm. So we've got the current, now we need some hard substrate and then we'll get more lost souls.
More
Or
lost souls.
poor souls.
Is this
Saw the ship move over.
Yep, the ship move is complete.
Yeah, it is.
what's that white sticky thing on that rock?
Yeah, let's go for zoom.
Go
I
for
think it's a
zoom.
bamboo coral.
Okay, I'm going to leave it for a sec.
I think you're getting like slightly pulled around by Atalanta's.
Yeah, I
Heat,
know.
so-
I got a little
And
little
then
ahead
is
of
that
you.
a
There's a little bit
sponge
still of
beyond that
settling
as
from
well?
Atalanta.
Come down a little bit. But
Are those some of
So
those little Q-tip sponges as well behind that?
No, those are actually barnacles.
Really?
Yeah, those little orange. Can you zoom more, Samantha?
Yeah, totally.
The yellow, those are barnacles. They're gooseneck barnacles.
really,
Oh, they're on the sediment
Oh.
beyond. Yeah.
Yeah.
Ah.
Megan, do you want to keep up this speed? Do you feel comfortable with
Yeah, I
Okay.
feel comfortable. We're going over some sand now, so we're probably going to go
Mhm.
fast anyway.
it? Okay. Bridge, Nav.
Is that sea star?
A 30-meter move,
Killing that
045, going
The brittle
0.3
star?
knots.
Yeah.
No, that's a dead sponge dock.
Oh, okay.
Yeah, so no.
So
They
it's
just like to hug things.
fair.
It feels good.
Like a potato bear.
Yeah, like a potato bear.
I'm getting to the point in
We
the
can zoom
cruise
out.
where I need a hug,
Zoom
I
it out.
think.
Yeah.
Aw.
Got a question. What is the ratio of regulars versus first timers on an expedition?
Oh, that's a good one.
It's always a little
Well,
different.
Yeah.
But Nautilus is like the OET does have a principle of like bringing out interns for a lot of the different seats and giving people their first opportunity to get out to sea. So
And then have
There
guest
we have it.
educators as
Yeah.
well as guest researchers
Yeah.
on board. So this is my first time.
And
Yeah,
it's
so you
my
got-
first time too.
Is this your first time on a research expedition or just your first time on the Nautilus?
On Nautilus.
On Nautilus.
On both for me.
Oh, nice.
I think it's another one of those sea pens with the sea star.
I love your commitment to those zooms that you're like, I'm going to set down and this is going to be.
Yeah, I get really close.
Yeah, one of the best ways to really get in there is to set down.
Yeah.
Especially on the
Practicing my
nice
sit-downs.
sandies.
And it starts, if you do, 100 of them, it stops being much of a commitment. You just
Yeah,
are constantly
like-
just gently bump something and use it to stabilize, and then you just move on. Setting down, getting up, moving around doesn't need to be a huge commitment, but it can be a huge pain if you have to get a sample in an awkward spot.
Yeah, there are some really awkward spots I've asked for, and I felt bad.
You shouldn't feel bad.
No, I know
You need to
love
keep
it.
us busy. Yeah.
with the video, should we ask video to push in a little bit just so we don't have the edge of the arm and the
Oh yeah, we can
you
go in a
probe
little
in
bit.
Yeah, I
there?
can also.
Do you-
A probe.
unless
The
you want
other
it
thing
for
off
frame
the
of
screen
reference.
was a dead sponge.
These ripples are really beautiful.
Yeah.
Aren't they?
Yeah.
They're very parallel. You don't have much fluctuation in that structure.
There is a lot of current up here, that's for sure.
Yeah.
When they're that elongate, that's because
Do you
there's
feel
no
it?
shortage of
Feel a
material.
little bit of it.
Yeah.
Sometimes you see little croissant
That's
shaped
not bad right now, let's say.
ripples instead, and that's where it's actually starved of sediment.
Yeah.
How deep would you think the sediment layer is here?
Don't know.
It's not that deep right here. Maybe an inch or two.
So pretty.
Yeah, so pretty.
And these are pretty symmetrical as well, so that suggests that the tide switches backwards and forwards. The current is actually flowing across kind of equal amounts left to right and right to left.
Go
If
for
they
zoom.
had if they were more lopsided, with one side being a slope and the other one being the sharp drop off,
Oh, okay.
then you'd be able to say, okay, it's permanently blowing this way, not the other way.
Yep. All right.
So if you were like in a desert under like the same kind of tropical winds that we have,
Yeah, when you get like
then you get like that, we pretty much get the wind coming out of the southeast all day, every day here with the trade winds. But down here, the stuff must be sloshing
Do you want me to
backwards
tilt
and
down a little
forwards.
for you?
Oh
okay.
yeah, a little bit of down might be helpful.
That's a snake star?
Yep.
Yes. 1
Nice
point.
one, Belle.
Nice one.
1 point for Belle.
Yay.
Okay, we can zoom out.
Okay, zoom it out.
It should be like little bingo cards of,
Yeah.
you've seen one snake star, one brittle star, one bubble vent.
We did do a bingo card on one cruise, but it had more interesting things on there, like, 1,000 boobies on the back deck.
Oh.
Okay, I can put another move in.
Yes, let's keep going.
Bridge, nav. Let's get another one of the same moves. 30 meters, 045, going 0.3.
That was nicely done, Gabby.
Wow, it's so pretty.
It's so pretty. I mean, I want more, I want some umbellulas, but
Yeah, me too.
oh, there's fish. There's something. What is it?
I just realized that I'm currently clicking 2 pens at
Oh.
the same time.
Oh, it's a jelly.
Oh, just
Jelly.
a little tiny
It's
one.
just blooping. You gonna go
Hey,
for zoom?
bud.
Going for zoom?
Is that what jellies do? They bloop?
They just bloop.
Yeah,
Look how
yeah.
cute it is. I thought it was a
Wow.
fish. I saw the
that's
shadow,
awesome.
Oh, that's so
but it's like,
cool. Is it upside down?
yeah, it's upside down. So this is like an inverted
Nice
one.
job on the zoom, by the way. These are not trivial to
Great zoom.
follow. Both video and pilot.
Teamwork.
Oh, yeah.
Wow, nice job.
Megan, you're crushing the following on this
Yeah,
one.
yeah.
I've always wanted to do this.
Megan's flying a camera. That's what she's flying.
Do you know what kind of jellyfish this is?
I think it's in the family Atolidae. I think it's got 4 tentacles.
She's still going.
Wow.
Yeah, we still have it.
Nice job. For the record, I have no idea how she's doing this.
Hyperfocus.
We're like so zoomed in. This thing is tiny.
So cool.
Well, you know, we've got some time here.
It's tired now.
All right.
It keeps running into the bottom. It's really
Yeah,
weird.
usually don't do that. All right, we can zoom
It's
out.
exhausted.
Nice
Never
job.
had to run that far to get away from the zoom.
All right.
Next victim.
How are gauges looking, Rye?
They are good. Sorry, you caught me right in the middle of a yawn.
That was cruel of me.
That's okay.
Probably ruined the yawn.
Let's see.
But yeah, I'll
I
do
think it's
another
a
gauge.
healthy sign. There's nothing alarming on the gauges.
Yeah, totally. She would not be yawning if there's
No,
something going on
no.
there.
What's that slight choking noise that you heard? Isn't me?
This is interesting. A little mixing it up. Lots of dead sponge. Oh, there's some stuff
There's some
coming
nice
up.
sponges coming on up, on upper.
Yeah, I see it. There's another C pen.
Well, it should just get increasingly rocky for the rest of the watch.
Hopefully. You never know. There's a mysid opossum shrimp.
Does the bridge typically change the heading without letting you know?
They're supposed to let you know,
Okay.
especially if it's more than a couple degrees.
Okay, it looks like it may only be 5 degrees.
Yeah, that should be fine, but I do appreciate when they tell me.
Bridge, nav? Did you put in a heading change? Okay, can you just let the navigator know when you do a heading change? Thank you so much. And we can have another move, 30 meters, 045 at 0.3. Sorry, make it 0.2. Beautiful, thank you. I'm slowing us down.
Thanks. Now that we got rocks,
Yeah.
we might want to look at stuff.
Yeah. Yeah, he just changed 5 degrees to starboard.
Okay.
It didn't look big, I just,
It's usually okay,
yeah.
but it's always a little disconcerting when you look over and your heading is changing and you're
Yeah.
not sure what's
I mean,
going on.
I'm surprised that he's kept this heading for this long because our forces are lined up
Yeah.
in a totally different direction.
But yeah, something under 5 degrees won't really make a lot of difference for us. More Boasoma.
Megan, I have a question here. It's how old do these sponge skeletons have to be to turn black? And is it manganese crust or natural face of death?
When it turns black like that, that's the manganese crusting. So you'd have to ask a geologist how long that takes.
We luckily have one on hand.
Some
Go for zoom.
thousands of years, I think.
Look at the
The-
shelf yellow.
The deposition rate, you can kind of reconstruct history like on manganese crusts in the ocean. You can sort of scrape a millimeter at a time, and you can go back millions of years through the history of the Pacific Ocean's composition in just like a few millimeters. So
Can we focus on the
really,
little
really slow.
guy that's on the sponge?
Yeah, totally. Yeah, we're pulling a little black.
Thanks.
Do you want camera tilt, Megan?
Yeah, sure. So this is a little benthic ctenophore on this sponge called Chompula.
Say Count Chompula?
Count Chompula, yes.
All right.
That's what it looks like to me.
I'm going to record that.
It's like spelled with a TJ. I'm kidding.
And what is it?
It's a benthic ctenophore.
Spelled with a CT.
Yeah, it's spelled with a CT.
Yeah,
Okay, we can zoom out.
the R is silent.
Very pretty. I like these. They're really cool. The lower is very blurry right now.
Yeah, I was trying to get a focus on that one that we
Ah,
were in front
okay.
of, but gone back to autofocus.
Cool.
And Gabby, there's going to be no need to stop at 70. We can just pass through and keep going to 8, which is going to be up to our northwest.
Okay. How do computers work?
Oh, yeah, HyperX is kind of annoying.
Uh-oh.
Oh, yeah. If you do the little box magnifying, you can like draw a box of where you want to be.
Can I center on the ship?
Yeah, there should be a center on the ship option.
Okay.
Probably
Under tools, maybe?
range tracking. Oh, I can't remember where it is.
Why don't you just stretch out a little bit in front, and then you can help me.
What'd you get?
I
See
could
just
also use
above
the
where you were
DVL
clicking?
reset too.
What's that?
If the next line up, Gabby, there's one called tools.
Yep.
Let's try that.
Tracking, none. Here we go. Nope, tracking in center, that's not helpful.
This one's fun.
How about view?
Oh, it's just quite slow. Yeah, I like view. Tracking and orientation. Tracking, vessel and
Yeah.
target. Here we go.
No. Okay, so click this guy.
This one?
Yeah. And then you can draw a box right around in here where we are.
Okay.
And I'll zoom in.
I had no idea this was where we were.
Yeah. And then
Are we here?
we should be here, I think. And just keep drawing lines.
Okay. Okay, why don't you pull ahead a little bit more?
Okay.
Are you sure this is the one we're
Oh,
at?
is that the one we're at?
I don't think so.
You kind of somehow rotated the screen.
I bet I did. Okay.
Yeah, that was samples 195, 196, it says. We're on 225, so
Yep.
what's the little red line down bottom left of that screen? Is that... That's not us either, right?
That it looks like a sentry line, a survey line of some kind.
Ah, okay.
Oh, yeah, that line is one of our mapping lines.
Is what?
It was a mapping line,
Okay.
so that's why I thought, since it was pointing toward that seamount, I thought that was
Okay.
the last one we did.
I think we're here.
Oh, yeah, that might be it. Yeah, that's it.
Oh, yeah. And
Now, I
and we
just
do
wanna
have
go
a rotated
north up.
view.
How about north up?
North up.
So old-fashioned.
You know, it gets the job done.
Yeah, there we go. That's better.
Good job.
You found us.
Yeah.
It was an
Even after we turned the waypoints off, she still managed to find us.
They'll never find us.
You really thought you could escape, I know it.
Yeah, we were making a break for it.
Okay, I want to look at this coral. Just take a zoom.
Can you do that again just when we do the watch handover, Gabby? Just like 30 seconds when they walk in. Say, okay, if you're the Gabby Nav, you should be able to figure this out.
Yeah. the Congress of
The
Gabbies.
real Gabby
The
Nav.
real
is
Gabby
coming
Nav,
up.
yeah, the real Gabby Nav, please come on watch.
Yeah.
This is a
Save
nice
us from
one.
the imposter.
It's very, very thin, but this is a bamboo coral. Oop, and it's closing its polyps. Very nice. Okay, we can zoom out. Can I get a DVL reset?
Sure. How do you do
Just
it?
send reset. And then
Oh,
say
and it
okay.
automatically is just going to go to USBL recent position?
Yeah, it's whatever is selected in that menu.
Yep, that's Megan
Thanks.
sitting on the Hercules seat.
That's a Chrysogorgia.
Okay. I'm going to work on, okay, so if I don't want to screw this up again. You use the arrow for doing range and bearing?
Yeah.
We still have 50 meters. So
You wanna
you
call
don't
in
want to
another
worry
move?
about, sorry, Chris, we got all distracted. You don't want to worry about going to 7, you just want to go straight to 8?
No, let's pass through 7, but
Okay.
but once you've put in the move for 7 and it's complete, we can just move on straight
Okay,
away.
great. We don't need to hang out there.
Not exactly, yeah.
Got it. Okay. Bridge, nav. Can I have a 40-meter move, 045, going 0.2 knots?
Yeah, we've got about 90 minutes left in the dive, so I think it's reasonable to try and get to 8 and then get Adam a rock from the top of the hill to go with the one at the base of the hill. And in the meantime, we'll have had the chance to see everything there is to see all the way out one side of the mountain. Looks like there might be a little bit
If
of
you
a
look at
saddle.
yourself in Atalanta, what do you notice about the vehicle right now?
I'm turned. And,
Anything else?
And I'm not quite in the middle.
Uh-huh.
I'm getting there.
I'm noticing mostly your altitude. Is that you're quite high up.
I am high.
And you'll be able to see more if you come down. You're very able to manage your position in the window now. So, you know, if you want to turn and get off to a side to like go look at something, that's what it's about. But you'll see it better if you're closer to the bottom. And that's Atalanta is another view that can help you figure out your altitude when you're on something steeper.
Would one of you be able to tell me, I don't see the depth so well here, is the summit, the highest point in the summit waypoint 7 or waypoint 8?
I think it's
waypoint
8.
Yeah.
Waypoint 8, okay.
Yeah.
So-
There's an extra 10-meter contour we're going to go up.
What
to get
is
to
that?
8.
A
It's
shrimp?
a shrimp. It's a Cantha fire.
So I think in that case, we'll want to fire 2 Niskins at waypoint 8, probably. I'm just checking with Annette to make sure she's also happy with that.
Yeah.
So we'll get 2 at the summit.
It's cute.
That's so
It's just
cool.
giving you
Are
a
they the
show.
last 2 Niskins?
Yeah.
So
Well,
no
the-
Niskins are
we
going
call 'em
to
the
have
drunken
been fired
shrimp.
yet.
This
We're
is
going to
like-
get 2 at the summit. 2 at 50-meter
Time to be
altitude.
like, "Do, do, do,
Oh,
do,
yeah.
do, do,
Ah,
do,
no,
do."
sorry.
vertical
Let me
profile.
just check. We're not getting any at the summit.
It's all going to be up in the water column.
So we're going to get one. We'll get them at the recovery in that case.
Okay.
So we'll get 2 at 50-meter depth. 50-meter altitude. 2 at a CO2 peak we saw in the water column, and then 2 at 50-meter depth. But I
Alright,
think that will be the
that's-
watch after us.
Yeah, exactly. I think we'll get as close as we can to the summit, and then they'll have an hour for
I love
sampling
those little
opportunities before they start
pleopods.
the recovery.
Love his yellow eyes.
Yeah. Well, the eyes aren't actually yellow. That's the light we're reflecting. All right, we can zoom out. Zoom out. Let him go.
It's interesting, we came across that sediment saddle. There's nothing like the density of filter feeders on this side of this rocky hill compared to what there was on the one we went down. Maybe it's, I wonder if it's linked to the, like, just how steep that topography was.
Yeah,
Maybe
maybe.
more.
Oh, there's a bunch of stuff coming up on the sonar for Atalanta right now.
There's a rock right in front of me.
Oh, it's got a lot on it as well.
That one's opposite banana.
Another Ramillo Gorge. Yeah, these are very common. It's definitely the land of brachyngeids and bolosomas. That's a pretty one. Sea cucumber. Go for zoom.
Going for zoom.
Yep, little sea cucumber. It's either an Emperima or a Peniagony. It's got a little vellum, that little hood. Very cute.
Would he be squishy if you chewed on?
Yeah, it would be a very interesting mouthfeel. We can zoom out.
Okay.
Be like a gummy candy, but mostly like water.
Like a gusher.
Yeah. When you dissect these things, they definitely do that.
Does
Yeah, if
anything
you
live?
right click and drag, you can measure.
Does anything live inside the holes on the sponges?
A lot of times there's like brittle stars and shrimps and
Is that where the
snails
wedding shrimp
and
are?
things that live there, but I haven't seen a lot of these sponges with associates.
Yeah,
And then
they
we
seem
saw
very empty.
that benthic ctenophore.
Bridge, Nav.
If we do see any with little snails,
Can we have a
Yeah.
20-meter move, 045, going 0.2 knots?
Megan, I'm gonna switch your starboard cam back to bucket and just see
Okay.
if that starfish is
Is in there, yeah.
feeling it.
That one was a cool one. I really hope it.
is this going to be our last move to get us to waypoint 7, Captain?
Yeah, pretty much.
This is nice caliphacus.
So I guess actually what we will want to do is
Sometimes these caliphacuses have the snails.
Ryan and Megan the chance to line back up, right? Change their orientation before
Yeah,
we do the next
exactly.
move.
Is that him in the corner of the flush jar? I feel like.
Oh yeah, I think so. I
Got
see.
him.
Oh, awesome.
Where?
It hasn't
See
escaped.
the little arm on the upper
Oh,
corner?
yep, yep.
Yeah.
So let's
Right.
switch off the flush jar. Like rotate to 7.
Yeah. Okay. I will put a note in the book that
Did you get that data?
starfish is in.
Got it?
15 minutes ago, huh?
Yeah.
He's been sitting there quietly like, I know where it is.
You already knew?
Yeah, he was making sure he didn't crawl out. He didn't want to distract you by asking you to rotate. I'm bluffing.
Oh, okay. I was like, you should have told me.
Yeah.
Here I am writing in the red book. Once it goes in the red book, I can't take it back.
And if you don't write it in the red book, do we have to give the samples back?
Yeah.
Yes.
Is it like
Yeah,
catch
you
and
definitely.
release?
We have to go back
If it's
down
not
and
in-
put them
if
back
it's
where
not
we
in the red
found
book-
them.
Yeah, exactly. Yeah, it's like, well, we have the navigation to do that.
Yeah, it'll just take a while.
We ain't never going home.
So I'm going to note that they should not run flush in the flush jar because otherwise the rocks will.
Yeah.
Yeah.
Dot,
Now
dot,
7
dot.
is
Give it a
the
headache,
flush.
yeah. Well, all the chambers must be loaded now, right? In the
6 and
we have
7, right?
Yeah,
Yeah, 6 and 7
free.
are open still.
Are they both open?
Yep.
Yeah.
Huh. If you can't flush, can you still use a chamber?
Yep.
I'm really surprised that none of these sponges have the snails. Go for zoom.
How tiny are the snails?
They're like a centimeter.
Okay, so like not microscopic.
Yeah,
be
they're
really-
not microscopic.
shouldn't be able to
You
hide,
should be
right?
able to see them. They're like white. All right. There's like, so you can see the brittle star in it.
Yeah, yeah.
It's like really up in there.
Tiny.
Since you had that question of, are there things in the holes?
That one has a tiny, tiny hole thing. Or maybe it's just only just poking out.
Yeah. It's worked itself in there. See, it's right there. Okay, we can zoom out.
Okay, zooming out.
All right.
the honey in my coffee was in that last sip. That was vile.
Oh no. This Calificus fell over.
Hmm.
It was so tall.
Recent mortality.
Yep. It's still alive.
Would it have had the same base color, or would it always have been white?
Once it's like fully dead, it'll be brown like all the other dead sponge.
How old are these sponges?
Oh, I don't know. It's hard to date them because they don't have any like good carbon in their skeleton. They're all glass, right? So I think there's a shrimp in there.
Yeah, I was gonna say, it looks like there's something on the underside of the stalk.
All right. All right, can you take it away with the panatote?
Yeah. Let's get him in there.
Okay, go for zoom.
Going for zoom.
Usually there's a Lebius. The Lebius like to hang out in here.
Oh, look at him.
It's a very happy little home.
Well, it was.
I think the shrimp's okay with it for now.
Yeah.
A lot of times these animals, since they don't eat a lot, they can live a long time after they've fallen over.
Right, they
Yeah,
must
we
have
can zoom
like
out.
really, really slow metabolism, right?
So, you know, this could have happened yesterday or, you know, months ago.
I wonder if either of those bases right next to it are where it was attached.
Could have been.
So Chris, we're like 10 meters south of waypoint 7 now.
Okay.
Are you okay with calling that good, or do you want to actually?
Yeah, no, that's good.
Okay.
We got to pass through the waypoint.
Ramming
So-
well, you can just like spin out right, Megan.
Yeah.
Just do
Our-
like a motorcycle turn.
Oh, yeah.
Our next move, I think, will be 000.
Yeah.
Just gotta go north.
I would go, you see where the grid line is, the horizontal grid line, Gabby?
Yeah.
I would go 000 as far as there,
Yep. And then
and
make
then hang
a
a left for 8.
Yeah. Okay. Sounds good. I mean, that's a ways. That won't happen on our
How
watch.
far
It's
is it?
115 meters to that
Okay.
horizontal line. Okay.
Right. Ramming speed then.
Okay, so 000 is our next move.
All
So if you two want to get lined up, Herc
Roger.
due north of Atalanta, and Atalanta pointing 000.
000. Roger.
See, it passed right through it.
Oh, good one. Thank goodness.
It was really important.
Yeah, it was.
I was up there with getting Sage to 3,000 meters last night.
Yeah,
I'm
exactly.
glad Sage got to 3,000.
Yeah, it's a good job we took the time yesterday because they started at waypoint 2 today.
Yeah.
Yeah.
Would have missed
Yeah.
the opportunity.
Well, glad we also got that small section of eDNA pumping from the bottom then.
You guys
Yep.
did end up getting that sample, hey?
We did, it's in the fridge.
Oh, awesome.
So, just one sample.
Are you guys okay if I start the move?
Yeah, let's go.
Bridge nav.
But then we've got the rest of the transect today, right? From
Let's
waypoint
go
2 on
40
up, so-
meters.
Yeah, everything
000
has been collected from the bottom.
at 0.2 knots.
Would you feel comfortable at going 0.3, Gabby, or
Yeah,
is it
totally.
too uphill?
I think so. Megan,
Okay.
how do you feel?
I feel good.
Bridge, can you make that move at 0.3 knots, 0.3 knots, 000 in the direction, or for 40 meters? Thank you.
All right, go for zoom. There's something on this sponge. This is another one of those Aspidoscopulia. I've got some brittle stars on there. They might have the snails. Those little white dots are usually the snails. I can't tell. Ooh, there's a cool tubularid. That's a hydrozoan.
So I think we've had a request that if there are snails on the sponge, it would be good to collect it.
I can't confirm
yeah,
that
they're
those
pretty
dots are snails.
Hard to see.
They are hard to see. Usually they're a little more obvious than just a dot. So I'm going to say those are not
I
snails.
don't think
Okay.
snails either.
But it's a good picture. Okay, we can zoom out. I'll keep zooming sponges until we find them. This is a nice old one. Okay, let's zoom this bolosoma, see if there's any snails. There's something on, no, it's just a hole. I got excited. No, no snails, we can zoom out. I would really like to know what those are, so I don't have to just call them gastropod.
Atalanta's started moving.
Great. Oh, look at this mysid. Want to try to zoom it? Oh, here it comes. And it goes. Okay, we can zoom out.
Okay.
It got real up close and personal to the camera.
It did. That's the hard thing about the swimming things.
I'm going to
Sometimes
pan
they
you
swim
left
for
because you're not square right now.
Yeah, that's a...
There you go.
Thanks. There's a sea star. Go for zoom.
Going for zoom.
I think it might be similar to what we saw. No, this one's different. This one looks like a Ceramaster. It's another type of cookie star, but it's not like the one that we picked up. Okay. We can zoom out. More of these bathypathies. There's a lot of these bathypathies. seem to be very common here. It's a dead sponge. What's down there? Not much.
You're off. I should be on.
Looks like a sponge straight ahead in lower.
Oh, yeah. Well, that's got to be zoomed in. I see it, though. Looks like another
Yeah,
Caliphacus.
It's mid-zoom.
That's great. I was just like,
Yeah.
it seems a lot closer than what I'm reckoning it is.
Yeah, I was looking at it through my rearview mirror.
Ah,
Bridge, nav. Can we have another move? 40 meters, 000, going 0.3 knots.
go for zoom. See if there's any snails. Oh, that's a nice texture, though. No, I don't see
If I was a
anything.
snail, I'd want to live on that.
Right? It looks like a perfect place.
Just looks,
So fluffy.
it does look like it, doesn't it?
It looks like a potato, like a fluffy potato.
It does.
You can zoom
Like a
out.
potato bear?
It could be, it does. It could be the mascot.
It could be the best.
That's what
It
the
kind of looks like a
Caliphacus.
shower sponge, really.
Mm.
It
Yeah?
does, doesn't it?
Yeah.
But I wouldn't want to use
It's a-
that.
it's a
That would
potato
be
bear
very
liver.
bad.
It would be a little soft, but
It,
you know, with the risk of,
Well, it's like it's
of glass
Glass.
splinters.
Good. Yeah, it's exfoliation.
exfoliation. I mean, like, maybe.
Is that a dead sponge stalk again?
Yeah, it's a sponge stalk. I was hoping it was an acorn worm, but alas, no acorn worms for us yet. I really like the acorn worms. They're really weird.
Why are they called acorn worms?
I don't know. I didn't choose that name, but maybe because I didn't want to call it what it actually looks like.
Which is the opposite of an
I
acorn.
thought they're also
Yeah.
called Yoda worms.
Yeah, some of them are called the Yoda worms. Yoda is a genus of acorn worm because the person who named it thought that it had ears that look like Yoda.
Hmm.
I
Was it
support- I
green?
support that.
Oh, it was purple.
Oh,
It's called Yoda purpurata.
purple yada.
Yeah, Yoda purpurata, which I think is a great name. But there are a lot of really interesting species names. There's a parasitic isopod, I believe, that is named after the killing curse in Harry Potter.
Hmm.
Because we're all nerds here.
Yeah, we should get Gabby Nav to relieve Megan and rig Tito out.
I bet Gabby would be a very good pilot.
Yeah.
Go for zoom. Look at that nomadic Ursinus. Got those really long legs.
Legs.
And a really nice Romilogorgia militaris.
Militaris?
Yep.
Like military?
Yeah, because all the branches are lined up in one.
Ah, okay.
Okay, we can zoom out. Okay. There's another one of those benthic ctenophores on this sponge.
Is there something on the edge of that one?
Yeah, it's a benthic ctenophore.
Oh, you just said that, sorry.
Okay. Do I put it in auto XY or?
I usually click all 3 autos at the same time.
Oh, okay. Like.
Whenever you put it in auto out, check your verts and make sure they didn't just pin.
Okay.
Actually, can you do me a favor and back away from this sponge? So right now you're in autos, so give yourself out of release XY. Back away from the sponge a little bit because autos do tend to drift in front. Like sort of takes very lightly.
Okay.
So that's great. Now you can auto XY yourself, and that's a great place to head over from.
Great.
Thanks. You can take off. Stay the next one over at the station.
Oh.
Okay, front row, when you're settled in, we're still waiting on Santiago, but if you're ready for another ship move, that'll be good just to keep going. The ambition is to get to waypoint 8 and the top of the hill if we can before the dive gets called on us.
Copy. All right.
And coming up hard.
Tito, I'm
what
going to
direction?
have you move in heading 350.
350.
Bridge nav. Can we step 30 meters in heading 350? Thank you.
And Gabby True Nav.
Yeah, right?
Gabby Apprentice Nav had us going up 0.3, just so, in case she didn't tell you. So I think that's what the ship will be doing is
0.3?
0.3, yeah.
Okay, thank you for that. We're flying.
Yep.
A whole 0.3.
Well, we got that updated recovery time quite early in our watch, so we just started cranking to see. We've been trying not to stop. The one thing that we have been on the lookout for, that Val and people ashore was interested in, is if we see any sponges that have a couple of tiny snails associated with them. That was something that people were interested in. So if we do get a chance to go look at some snails and zoom on them, but that might be something to wait till Santiago's here, because he'd actually know what he's looking for. We were kind of relying on Megan as our biology guru.
Yeah, Megan is definitely the biologist of this watch.
Yeah. Actually, can we zoom on that sponge there, Tito?
Sure.
They said they were like little white snails are the things we're looking for, apparently.
I do see something white on that.
Yeah, that's what I was thinking. There's this little thing just there.
Do you want me to hold the ship?
No, I don't think we'll be here that long.
Copy.
Yeah, I think that's...
We just want to see what's inside that honeycomb structure. Nothing that I can positively identify as a snail.
It looks just like
Yeah, okay, thank you.
other sponge.
Let's keep cranking.
Looks like a pair of them.
Yeah.
We've had these for the whole dive, just
Yeah.
like forests of them.
Wow.
Not quite forests, sparse forests.
Well, there was one part when we came over that cliff where there was a lot of filter feeders.
Yeah, there was
There
a
was
lot.
a lot there. Did make me think of a wind farm.
Yeah, yeah. They're really beautiful.
Oh, Michael, just a heads up. So the flush jar and the slurp, yep, you know? Okay. Perfect.
Ooh. There's a lot of them here.
Mm. Isn't that pretty?
And this one closest to us on the right is a different species, but I don't know what it is.
That one in top right, you think?
Yeah,
Back
it
up
hasn't
to
got
it.
the same yellow stem. It's got this much longer...
That one there.
Yes, sorry. I don't have any annotations on my screen.
I could have
Ah.
sworn I heard left. Yeah.
Had you said left for Mary or right?
No, it was the one Chris circled, the right.
Oh, it was when I
Yeah.
cycled. Okay.
Yeah.
Over this direction.
With a really long little
Long
stem.
It has got a red thing on the bottom corner of it. Maybe a brittle star.
Ship is coming to a hold.
We could keep moving.
Copy.
They seem to be
Yeah, it's
trying
just
to make some
one of
time.
those
Oh, what
little
is
brittle
that?
stars at the bottom of the stem.
Bridge nav.
Down there, you mean?
It's an old stock with a
Orioles.
crinoid around it.
Can we step 30 meters, heading
push.
350?
It's just around one of the old stalks. Oh, yeah, it's multiple dead stalks with crinoids wrapped around them.
Thank you.
Moving on. So what are you thinking for time off bottom, Tito, if we got recovery at 1900?
2,400 meters, about 5 o'clock.
Yeah.
And do you know what time the next dive will be after that?
I think Derek and Adam have told me that the plan was that we do the transit and then it's going to be Sentry first before
Okay.
the ROV. So I think that might put Sentry in around midnight.
Yep.
And if they're going to be one after the other rather than in parallel, then Sentry is probably another 6-hour dive for
Yeah, that
Guangyu.
sounds right.
So then ROV dive will be in before breakfast tomorrow.
And they are going to be sequential, or you aren't sure?
I haven't looked at the whiteboard in 4 hours.
Yeah,
so
me
No,
neither.
it's not been updated.
who knows?
quite
Okay,
yet,
okay.
So,
But I think all the ROV, all it said so far was just we'd
Transit,
be leaving
yeah.
and transit.
I wasn't
but I-
sure if...
At the watch changeover, I asked Adam what he was thinking as first operation, and he said Sentry.
okay.
So I know I was texting Guangyu before I came on watch about ideas of how he's going to set up his final dive.
Yeah.
But I said I'd go check in with Sean when I get out of here and see if they've closed the loop on that.
We're heading back to Vaialulu'u.
Yeah. So we, Guangyu's project seemed to work pretty well yesterday, in that he was flying with a new sonar on Sentry over the same place where we were doing the photogrammetry the other night on the bubble vent.
Oh.
So he was able to fly over the same things and show that he could actually measure it from 25 meters up. He could see it in the multi-beam. So pretty much same as Norbit can do, but covering a lot more ground, a lot faster.
Wow.
So, but as part of that survey, what he also found was that his survey went a bit further afield than the survey that Mary and I had been involved with earlier this week. And there was evidence for another bubble plume that was off about 200 meters east of the bubble plume in a place where we haven't been yet. So I think his new survey is going to plan to go back through there and see if we can track down more evidence for more bubble plumes on the outside of the crater wall. And if everything works out absolutely marvelously, then we can persuade Val that of all the different ways she could drive up the outside of Vaialulu'u to look at the biology on the outside of the crater, maybe her dive could just happen to end at the top of the crater on that eastern side, just to see if we can just find one more vent before we go home, or before we leave this area anyway. but that was the real driver for going back, was actually Val and the sanctuary would really like to know what the ordinary fauna are living on the seamount there on the outside, rather than living near where the active volcanism and the hydrothermal venting are.
You can also get some gas emissions from the outside of the cone sometimes, so it'll be very interesting to see whether we get any CO2 signature, not either on the rim or within the crater, but coming from the sides of the mount. And I don't think that's something that's been studied at this site before.
Bridge nav. Can we step 50 meters, heading 350?
And of course I did hear, I was listening into one of this watch's broadcasts earlier in the cruise at Vailulu'u, and I remember listening to Tito saying how much that the low temperature microbial map with the low temperature flow inside the crater reminded him of stuff he's seen on the outside of the seamount in Hawaii that was down at around 4,000 meters on the outer flank. So you never know, there might be interesting stuff we're not ready for on the outside as well.
I think the outside will be super
Hey,
interesting,
Santiago.
just for the contrast, even if... Yeah.
I figured you were probably getting excited about something. So, are you up to speed with the fact that Guang Yu has found evidence for a new bubble plume east of the bubble vents we've already been to? Or do you need to go back down and reconsider where you're going to go with your dive? Yep. So you're going to climb the wall on the outside and maybe end up there at the top. Okay. Well, I think Guang Yu's actually coming up with a survey that's going to happen before your dive starts as well. That'll carry on extending that search out. So.
For those of you asking, I think we probably have about 3 hours left in this dive, give or take.
Yeah, but only an hour of that is going to be on the seafloor, I'm afraid.
Yes, but they get to listen to us for 3 more hours.
Yeah, you get to listen to Blue Radio.
Right.
Drive time.
And then we transit to Wailu'u, so...
Yep.
And I know some of you are asking about a dive schedule. We don't share a dive schedule in advance, but the best way to stay updated about when we're diving is to subscribe to our Twitter or Instagram or Facebook, where we do updates, or of course just to check in on the website and check at the status update.
I can't even keep track of our dive schedule.
No, which is why we don't share it, because we don't know ourselves.
Yeah.
We've been so fortunate this trip with knock on wood, but we've had good weather compared to the last cruise.
That is very true.
Yeah.
Have we gotten a Niskin yet? I don't know if the first one's been pulled or not.
It doesn't look like any of them have been pulled.
I think we
Well,
want
the
to
first
do the
one is
Niskins
like in
in
the way of the
waterfall.
like behind the camera, kinda, but I think it's right there.
What did you say in that?
I think we were saving the
I saw
Niskins for the water column.
That's what I heard as well.
That,
The water column?
Yeah.
Yeah.
Okay.
So the star-shaped things that we're seeing on stalks, are they crinoids or are they brittle stars? Or are there both? Does anyone know? There's no biologist.
I believe we have a crinoid over to the right.
Okay.
And also wrapped around the stalks. And then we have a nice coral right in front of us. Jaina, would you push? Okay. Sorry, lost it a little there. Pan right a little. And go ahead and push.
Oh, look at that.
It's one of those dandelion-looking corals.
And let's pull back out, please.
I mean, yeah.
And want to give it a quick push. Trying to come down a little.
All right, so we are right now we're making moves to waypoint 8, correct?
Yes, we are.
And
Okay.
pull back out.
Sounds good.
We're trekking. We're actually going, you know, up to 0.3 knots.
Okay.
So, not
And
messed
what
around.
time do we have to get off bottom?
At 5, so wait about
At
what,
5?
At 5-ish.
Top of the hour.
Okay. So we have 45 minutes. Seems like there's a lot less life right at the summit.
Tito, I'm going to have you start moving in direction 310.
Roger that. Oops.
Bridge nav. Can we step 30 meters, heading 310? Tito, can I do a quick DVL reset?
Absolutely.
Thank you.
What type of rocks do we see around here? The traditional ones?
The traditional.
I guess that was a weird way to phrase it.
Classic rocks.
Lots of...
Oh, go ahead.
Lots of like old pillow basalts and just basalt rocks in general. Yeah.
But this is an old seamount, so pretty much every rock that you see is coated in iron manganese crust. But that's why they look so dark and cemented. So you see there's not really loose rocks, they're all glued together by that crust. And now we're seeing a lot more sediment because we're reaching the summit and it's more like a flat spot. So sediment can accumulate instead of just washing down. Although it seems rubbly here. Is that my impression? It looks like... Can we zoom in on the sediment, Tito? Or are we on a fast move right now?
Sure, I'm a little out ahead. Coming up to a wall here, though. We'll have to get a little sporty in a minute. I'm going to go ahead and land here.
Parkour.
Parkour.
Let's go ahead and push.
Ooh.
Definitely rocks.
Definitely. Well,
Astute
the key is that they're
observation.
Small rocks. So, Jacob, where are those coming from?
Could be from mass wasting, maybe parts of the
And right back out, please.
wall above us that are just weaker from faults and fractures that are kind of crumbling off and weathering. Could also, I don't think they're nodules, so that wouldn't make.
They don't. No.
They don't look like them.
They look like it has sharp angles, but they're all small, very small, right?
We've reached the wall.
315.
It's a very short
Let's
wall.
see exactly. Yep, I have the ship going 315.
Yeah,
So we
so
can
you
proceed
see
in
there,
that direction.
Like, chunks of the wall. Ooh, what's that on the left? What's this? You see that?
Oh,
It looks like a pipe.
a pipe. Huh.
Oh, it's a dead sponge, I think.
Is it?
Not sure.
Yeah, looks like a sponge vertebrae we sampled this morning.
Right, yeah. A fossilized sponge.
Hmm.
This one is very dark. It's been here a long
And
time.
let you push, please.
I didn't realize that sponges could fossilize.
Yeah, we've seen a lot of those on at these depths, and the couple Simons we've dove. So we dove in 2 Simons that was in the before in the southeastern part of the
Bridge
American
nav.
Samoan territory that
Restep 30 meters, heading
had a lot of dead sponges
315.
just like here, and some of them were on the way to being fossilized, like this one.
How does that happen?
Well, you see the dark? It's
Yeah.
also that iron manganese.
That's
crust.
ferromanganese crust? Oh. So it doesn't just form on rocks.
No, it can coat any material that I guess is laying around for long enough to.
Yeah. I guess I'm just surprised that a dead sponge didn't get eaten up.
Right. So it's probably because it has a pretty undigestible
Really?
siliceous skeleton.
Oh, okay.
So the organic tissue material probably is digested
Yeah.
early, but the
So they kind of have like glass bones
yes, that's right.
in a non-scientific
Very
way.
much, yeah. So we did pick up a piece at the beginning of the dive, one of those fossilized
Cool.
sponges. So we'll be able to take a look at how thick that crust is,
Oh, yeah.
and maybe if the spines that form the skeleton are still in decent shape, they could be identified. It's interesting how we were climbing the ridge and we got to the edge of the summit. There was a lot of corals and sponges. but here, it's very little.
Do you think it's because of like currents around the summit that might be?
Possibly. I mean, this is much less steep terrain, so it's possible or likely that the current acceleration over the terrain is less than at the ridge where it's steeper. So maybe it's just not fast enough to deliver enough food particles for sponges and corals to grow to be large and in high numbers. You still see, there's a few of the corals we've seen before. There's a bathypathes, and there's a couple of those Armuligorgia, but nothing large and very few sponges. Definitely not the bolosomas that we're seeing.
So can currents also like isolate parts of the seamount from like different larvae, or like prevent larvae from settling in different areas, or?
Possibly. I think it depends on the nature of the currents and the shape of the seamount too. So some seamounts, the more conical shape, can actually create, when they interact with the impinging current, they can create like a recirculation cell that stays above the seamount. So we call Taylor cones. So the idea is that maybe those act to retain both larvae and food particles over the seamount. So one of the, I guess, long-standing hypothesis is that maybe seamounts could be like semi-isolated habitats where basically larvae get released and they're not able to leave easily. I think over the years we've learned that that possibility of isolation leading to endemism in seamounts is not well supported by observations.
Tito will continue heading 305.
305.
Bridge nav.
It also depends on depths, right? Shallower seamounts.
We step
like
30 meters,
have
heading
tidal
305.
points, so they have tidal currents. Right. These ones that are so deep probably have much less tidal current influence. So the current regimes vary depending on the depth as well. It's more fossilized sponges. I see some tall sponges, living sponges coming out. Scalophlacus sponges. Oh, there's a coral.
It looks surprisingly leafy.
It does. Leafy? I thought so too.
Yeah.
The pattern.
Yes, it does. So we saw a huge one earlier this morning. It was like a 3-meter long sponge that was being fossilized like this one, but much larger. But then I think I haven't been sitting through the whole dive, but I think we also saw one living one that looked like that.
I think I saw that one.
You saw that one?
What kind of is that? What kind of sponge?
I don't remember the name.
That's okay. How dare you not know every single sponge?
It's the only one that we've seen alive, right? It's really interesting to see like things that look exactly the same being fossilized next to a living one. It's like tells you that that species has been living in the seamount for a long time.
Yeah, because we think of fossils, we think of animals that no longer exist.
Right.
But not down here.
And
wow.
the
And come back out just a wee bit.
the rate at which those iron manganese crusts accumulate is in the order of millions of years per milliliters. Millimeters.
It looks like a footprint.
Do you know if that rate varies depending on the region?
Oh, let's
I
pull
mean,
back
it,
up.
it would make sense if it would, because like in regions closer to like hydrothermal vents and like areas with like more mineral, higher, you know, concentrations of minerals in the water might experience faster rates of precipitation.
Yeah.
I'm not sure though.
That does make sense. Iron manganese are metals that are very commonly found on hydrothermal vent fluids. And we do have near hydrothermal vents nearby at Bailulu, so it's possible that maybe the seamount and other seamounts in the vicinity have faster accumulation of crust.
Also, we have a kind viewer who let me know that the sponge is a Farreidae. Am I pronouncing that right?
Yeah, that's the family, yes. I'm just not sure what the genus is or species. Yeah, Farreidae is the family.
Farreidae. I'll never get the hang of pronouncing Latin.
Well, it's a dead language, so I think you can't be wrong, right?
As long as you can read it, right?
Spiky thing on the ground.
An urchin.
That's interesting.
Oh, yeah.
Oh, yeah, that looks like an urchin.
That's first urchins we've seen down here.
And you want to push?
Oh, it actually looks like an anemone more.
Oh.
Or,
It's like a Pokemon.
It really
Pokemon.
does.
I'm actually not sure it's an anemone. It could be one of those. Two anemones or Sorrentarian. Couldn't get a close look, but maybe somebody in the chat recognizes it. The lone
Bridge
sponge.
F. We step 30 meters, heading 305.
We have a guess from our audience that it's a pom-pom anemone. I'll look that up.
I don't know the common name.
Oops.
I've called pom-pom anemones to other kinds of anemones.
Limponema? No, maybe. I do not know.
This guy's got a grip on the sediment.
Yeah.
But the sediment's got a grip on him.
Is that common for sponges to attach straight to the sediment?
No.
Huh. Good for you, sponge.
Well, it might be attached to a rock that just got covered by sediment.
Oh.
No, I don't. Well, there are some. I've seen some, but not like this one. They're mostly animals that live in hard substrates, like corals. Although there are corals that live in sediments, they're specialized to live in sediments. So they create not like glued attachments to the rocks, because obviously there's no rocks, but they can form structures that look like roots.
Oh.
It's interesting because this is more or less the top of the seamount?
Is it flat?
I think it is, I mean, for the cameras, it looks like it has a slight incline.
I guess I don't really understand what makes a guyot versus a seamount.
Guyots are flat top seamounts, but they usually have carbonate crust on top, so like a that is biologically formed, you know, by.
So the shape is different, but also other parts about the top are different.
Yeah, so the flat top, like a conical seamount, when it gets shallow enough, there can be growth of photosynthetic organisms that secrete calcium carbonate, like corals and algae. And over time, they kind of built a shelf on top of the seamount. So in fact, I think in a couple of days, we're going to be diving on examples of gills. When they're shallow enough, they call them banks. Although bank is just something that gets closer to the surface. So there are gills that are banks, but underlying there's a seamount. So some seamounts in navigation charts, they're just called banks.
So looks like there is some impressions in the sand there, in the sediments.
Like tracks?
Yeah, or maybe it's just the way the currents created it, but.
Yeah, it could be. I do see ripples that look like formed by currents. We're almost at the summit.
About 60 meters.
60?
Yep. Bridge nav. We step 30 meters, heading 305.
That's more dead sponges. Yeah, there's another balsama sponge. I haven't seen those in a while. So all these sediments likely have a biological origin. So it's from plankton remnants that rain down from the surface and accumulate. And that we know that because we're far away from any land here. So it's likely not mineral materials that are eroded from an island or something like that.
That is really cool looking.
The thing is a coral growing on this dead sponge.
Yep.
Let's see if it's actually growing on the sponge or on a rock through the sponge.
Okay, now we will push.
It looks like the base is on the sponge.
It certainly does.
Yeah. Interesting.
And then what's on it? There's something
Yeah,
crawling.
look at that.
I think I've seen corals growing on other corals, but never a coral growing on a sponge.
Yeah, what's the organism on the coral?
That's a squat lobster.
Squat
And
lobster?
it looks like a much smaller little coral off down and to the right.
This one?
Yep.
Yeah, good spot.
Oh yeah, you can see the base of that coral is on the sponge. That's really neat. I think that little one may be another sponge, like a carnivorous sponge, or doesn't look like a coral. It's very small.
Yeah, pull out, please. Atalanta's catching up to us.
Okay.
It's not a competition to the waypoint. Okay. But slow and steady does well. Some of these look blue. Like a very light blue. Or is that just me?
I can see it.
Some more bed sponges.
Bridge nav. Can we step 30 meters, heading 305.
I wonder what causes those sponges to die. You
They're
definitely
just
see
all
bigger
over the
ones.
place here. You can see
Yeah,
them.
yeah. They clearly can get much larger than this, right? 'Cause we saw huge ones. Wonder if they just get big enough that the currents can break them.
Oh, look at that.
We're at the summit. Like this. So there's more bolosoma sponges. Tito, can you take a look to the left? Thought I saw a big dead sponge. No, I think I was just imagining it. There's some large volosomas here on top. Gabby, do you know if this is the highest peak of the seamount?
It looks like it, according to this little topography I have here, or the bathymetry overlap.
I know that
Let me
this ridge
zoom out.
extends further to the northeast, but I don't remember if it gets any shallower or is everything downhill from here.
Let's see, this is 8. It gets shallower, but just by 30 meters.
Okay. So this is like a local summit, but it's one of the peaks of the top of the volcano, of the seamount. So we have about 20 minutes left. So I don't know if it's...
Do you want to just mosey around this area?
I think so. I don't think it's worth investing in another move.
We can hold the ship.
Maybe we just spend more time doing some close-ups on the sponges. I know they were seeing some snails earlier that people on shore were interested in finding and potentially sampling.
Would we like to trip any Niskins or anything?
So the plan for the Niskins is to fire 2 of them, so 1 and 2 at 50 meters above the summit. Another set of 2 at around a depth of 580, but we're gonna want to keep an eye on this CO2 sensor on Sage, and we'll tell you. We saw a CO2 anomaly around that depth, so we're interested in getting a sample of that water. And then a third set of 2 at a depth of 50 meters. So close to the surface.
Santiago, I have 560, not 580.
Yeah, as I said, I think we need to keep an eye on the Sage to inform that.
Okay.
It's a depth range.
Yeah. Okay.
Yeah, I think you're right. It's 560 when we saw the highest signal, but...
would it be possible to go more to the left?
Yes.
Only if we move the ship a little bit.
Yep, well, we can move the ship left.
Since we don't have too much time left, I'm just going to keep everything on this
I
pump.
think we can go, let's say, 275.
That'll give us an opportunity for 1 pump sample in the water column.
Bridge nav. Can we move 20 meters, heading 275?
So, Volante is going to want to go up there.
Because it's already going in that direction?
Yes.
Okay.
Can you tell if there's much of a current out here, Tito?
You know, let me go ahead and zero things up and put it in auto depth, and we'll do a little measurement.
There is.
Sounds great.
Because the...
Could you zoom way in, and we'll see which way it's going? Let's zoom way in on Herc.
There's a...
I'm sorry, I was talking to the navigator.
All right.
Oops. So I'm going to say it's going south. Maybe just east of south, but mostly south.
Going to south?
Yeah, I'm watching it on the nav screen here. Well, as I look closer, seems...
You can see the tether as well, kind of pushing.
Yeah, southeast.
Going to the southeast, okay.
What's that?
I say going to the southeast.
The current is going to the southeast, yeah.
Okay, thank you.
You're very welcome.
I can hold here. Bridge nav. Can we hold here?
Wow, you can see so many different
Shana, would you push on that?
stocks accumulated here. Makes you wonder how many generations of sponges that represents. You see the fossilized, different species, fossilized sponge underneath.
See over 10 pieces right there.
Yeah.
Yeah, come out a wee bit. I'm going to come down a little bit.
It's interesting is that,
to the right.
at least on this seamount, we don't see as many
good.
coral skeletons. Whereas in a couple of the other seamounts to the southeast, we did see a mix of both coral and sponge skeletons on the seafloor getting fossilized. Here, I don't think I've seen skeletons that look like coral. They're mostly sponges. And they're obviously the most dominant group on the seamount. Very abundant all the way from
So maybe the current will keep Atalanta down there a little bit.
Our
Seems
first
like it.
waypoint, which is waypoint 2, all the way to the summit. And that started at 2,700 meters, so we climbed about 400 meters vertical on this dive.
Yeah, would you push on that sponge? Yeah, back out, please.
Nice image. So we are facing the heading of the ROV is 160 degrees, so you can actually see the ripples.
It's about 290.
Oh, 290, sorry. I was looking at the heading of Atalanta, I think. 290, so that's...
Amazing how many dead sponges, just everywhere
Yeah.
you look. I've counted 35 or 40 within a couple square meters.
Do we see any tiny sponges anywhere? I'm not sponges, snails.
I did not see any on this zoom. Yeah, so we're facing to the northwest right now,
northwest, yeah.
and you can see that we are like our view is perpendicular to the ripples on the sand, on the sediment, and so that also is an indication that the current that we're facing right now is more constant over time than just the few minutes that we're able to spend over here.
There's more sponge carcasses over here.
Usually sea fan corals are a better indicator of the current, but there's none here.
That's interesting with that.
May I have a push?
Looks like some sort of Halloween costume.
It really does, doesn't it?
Jeez.
It's a new sponge growing on it. Oh no, it's just the top of a dead one.
Yeah, but it's very weird. It looks like it's, yeah, the bottom looks like it's dying, but the top looks healthy.
Looks okay on top, yeah.
Looks like somebody that showed the Last of Us.
Oh.
Or looks like the sponge is about to cosplay at an alien convention.
Yeah.
That's interesting. I wonder what it's causing that kind of slow. Maybe it's progressive.
That looks like an enormous piece right there.
Ooh, yeah. Let's see, so we sampled a couple of sponges, right? We got in our previous watch, we sampled one of those concave sponges. I forget the name. When we sampled, I think, a small...
Would you push, Jenna?
Bolosoma.